Introduction to gene editing. Gene editing refers to a group of technologies that give scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at particular locations in the genome. The precision and efficiency of modern gene editing tools have revolutionized biological research and opened new avenues for treating genetic diseases. Understanding the fundamental principles of these technologies is crucial for anyone entering the field of biotechnology or genetic medicine. This course will delve into the various methods and implications of modifying genetic code. It represents a significant leap forward from earlier, less precise genetic modification techniques. The ability to make targeted changes to DNA has profound implications across many scientific disciplines. Central Dogma Review. The central dogma of molecular biology describes the flow of genetic information within a biological system. It states that information flows from DNA to RNA, and then from RNA to protein. DNA serves as the stable blueprint, containing all the genetic instructions for an organism. RNA acts as an intermediary, carrying these instructions from the DNA in the nucleus to the ribosomes in the cytoplasm. Proteins are the functional molecules that carry out most of the work in cells and are required for the structure, function, and regulation of the body's tissues and organs. Gene editing directly targets the DNA, thereby influencing the subsequent RNA and protein production. Any alteration at the DNA level can have cascading effects on cellular processes and organismal traits. Basic genetics concepts. Understanding basic genetics is fundamental to appreciating gene editing. A gene is a basic unit of heredity, consisting of a specific sequence of DNA or RNA that codes for a functional product, typically a protein. Alleles are different forms of a gene, arising by mutation, and found at the same place on a chromosome. Mutations are changes in the DNA sequence, which can be beneficial, harmful, or have no effect. These genetic variations are the raw material for evolution and the basis for individual differences. Gene editing aims to precisely introduce or correct these mutations. The intricate interplay of genes and alleles determines an organism's traits and susceptibility to various conditions. Chromosomes, which are structures made of DNA and protein, house these genes. Early gene editing techniques. Before CRISPR-Cas9, scientists utilized other gene editing tools to modify DNA, albeit with less precision and efficiency. Two notable early techniques were zinc finger nucleases, ZFNs, and transcription activator-like effector nucleases, TALENs. ZFNs are artificial restriction enzymes generated by fusing a zinc finger DNA binding domain to a DNA cleavage domain. TALENs are similar, using a DNA binding domain derived from transcription activator like the effectors, tails fused to a nucleus. Both ZFNs and TALENs rely on custom-designed proteins to recognize and bind to specific DNA sequences, then cut the DNA. While effective, their design and synthesis were complex and time-consuming, limiting their widespread application. These early methods laid crucial groundwork for the development of more advanced gene editing platforms. They demonstrated the feasibility of targeted DNA modification, paving the way for future innovations. CRISPR-Cas9, Discovery and Mechanism. CRISPR-Cas9 is a revolutionary gene editing tool derived from a natural defense mechanism found in bacteria and archaea. It was discovered as part of the bacterial immune system, which uses CRISPR, clustered regularly interspaced short palindromic repeats, sequences, and Cas, CRISPR-associated proteins, to detect and destroy foreign DNA, such as from viruses. The mechanism involves a guide RNA molecule that directs the Cas9 enzyme to a specific DNA sequence. Once Cas9 locates its target, it acts like molecular scissors, making a precise cut in both strands of the DNA. This double-strand break then triggers the cell's natural repair mechanisms, which can be harnessed to introduce desired genetic changes. The simplicity and efficiency of CRISPR-Cas9 quickly propelled it to the forefront of gene editing research. Its discovery transformed the landscape of genetic engineering, making targeted DNA modification more accessible than ever before. Components of CRISPR-Cas9. The CRISPR-Cas9 system primarily consists of two key components, the Cas9 enzyme and a guide RNA, gRNA molecule. Cas9 is a nuclease, an enzyme capable of cutting DNA. It acts as the molecular scissor responsible for making the double-strand break at the targeted genomic location. The guide RNA is a synthetic RNA molecule that is engineered to have two parts, a CRISPR RNA, cRNA, sequence that is complementary to the target DNA, and a trans-activating cRNA, tracker RNA, sequence that binds to Cas9. The cRNA portion directs the Cas9 enzyme to the precise DNA sequence to be edited. Without the guide RNA, Cas9 would not know where to cut. Together, these two components form a powerful and highly specific gene editing complex. Their synergistic action allows for unprecedented control over genetic modifications. The design of the gRNA is critical for the success and specificity of any gene editing experiment. Targeting specific DNA sequences. The remarkable specificity of CRISPR-Cas9 largely depends on the design of the guide RNA. The cRNA portion of the guide RNA contains a sequence that is complementary to the target DNA sequence in the genome. This complementarity allows the guide RNA to bind to the DNA through Watson-Crick base pairing. For successful targeting, the target DNA sequence must also be immediately followed by a protospacer adjacent motif, PAM sequence. Typically NGG for Streptococcus pyogenes Cas9. The PAM sequence is crucial because Cas9 will only cut if it recognizes this motif, preventing the enzyme from cutting indiscriminately. Scientists carefully design the guide RNA to match the desired genomic location, ensuring precise gene editing. This meticulous design process minimizes off-target effects and maximizes the efficiency of the editing. The accuracy of the guide RNA is paramount for the success of any gene editing application. DNA repair pathways. Once the Cas9 enzyme makes a double-strand break in the DNA, the cell's natural repair mechanisms are activated. There are two primary pathways that cells use to repair these breaks: non-homologous end joining, NHEJ, and homology-directed repair, HDR. NHEJ is a quick and dirty repair mechanism that ligates the broken DNA ends back together. This process often results in small insertions or deletions, indels, at the repair site, which can disrupt gene function by causing frame shift mutations. HDR, on the other hand, is a more precise repair pathway that uses a homologous DNA template to accurately repair the break. Scientists can introduce a synthetic DNA template containing desired genetic changes, which the cell then uses for repair, leading to precise gene insertion or correction. The choice between NHEJ and HDR dictates the outcome of the gene editing process. Understanding these pathways is critical for designing effective gene editing strategies. NHEJ is more prevalent and active throughout the cell cycle, while HDR is primarily active during the S and G2 phases. Applications of CRISPR-Cas9. The versatility of CRISPR-Cas9 has led to a wide array of applications across various scientific fields. In basic research, it is extensively used to create knockout models, where specific genes are inactivated to study their function. It also enables the introduction of specific mutations to model human diseases in cell lines and animal models. Beyond fundamental research, CRISPR-Cas9 holds immense promise in therapeutic applications, offering potential cures for genetic disorders by correcting disease-causing mutations. Furthermore, it is being explored for developing new diagnostic tools and antiviral therapies. The ability to precisely manipulate genes has opened up unprecedented opportunities for scientific discovery and medical innovation. From understanding gene function to developing novel treatments, CRISPR-Cas9 is a powerful tool. Its impact is continually expanding as new applications are discovered and refined. Gene editing in agriculture. Gene editing offers transformative potential for agriculture, enabling the development of crops with enhanced traits and improved resilience. Scientists can use CRISPR-Cas9 to introduce specific genetic modifications that lead to increased yield, improved nutritional value, and enhanced resistance to pests and diseases. For example, gene editing has been used to create wheat varieties resistant to powdery mildew and soybeans with healthier oil profiles. It can also accelerate breeding processes by precisely introducing desirable traits that would otherwise take many generations through traditional breeding. This technology provides a more targeted and efficient approach compared to conventional genetic modification, GMOs, which often involves introducing foreign DNA. The precision of gene editing allows for subtle changes that mimic natural mutations, potentially easing regulatory hurdles. Ultimately, gene editing in agriculture aims to address global food security challenges and reduce the environmental impact of farming. Therapeutic gene editing. One of the most exciting applications of gene editing is its potential to treat and cure genetic diseases. By precisely correcting disease-causing mutations in a patient's DNA, therapeutic gene editing offers a permanent solution rather than just managing symptoms. Clinical trials are underway for various conditions, including sickle cell disease, beta thalassemia, and certain forms of blindness. The approach involves either editing cells outside the body, ex vivo, and then reintroducing them, or delivering the gene editing components directly into the body, in vivo. This technology holds the promise of transforming medicine, offering hope to millions affected by inherited disorders. The ability to fix genetic errors at their source represents a paradigm shift in treatment strategies. Ethical considerations are paramount in these applications, ensuring patient safety and responsible use. The long-term goal is to make these therapies widely accessible and affordable. Ex vivo versus in vivo gene editing. Therapeutic gene editing strategies can be broadly categorized into ex vivo and in vivo approaches, each with distinct advantages and challenges. Ex vivo gene editing involves removing cells from a patient's body, genetically modifying them in a laboratory setting, and then reintroducing the edited cells back into the patient. This method allows for precise control over the editing process and rigorous quality control before reinfusion. It is commonly used for blood disorders where hematopoietic stem cells can be easily accessed and manipulated. In contrast, in vivo gene editing delivers the gene editing components directly into the patient's body to modify cells within their natural environment. This approach is more challenging due to delivery hurdles and potential off-target effects, but is necessary for tissues that cannot be easily removed or transplanted. Both methods aim to achieve therapeutic outcomes but employ different logistical and technical strategies. The choice between ex vivo and in vivo depends on the specific disease, target cells, and safety considerations. Each approach presents unique opportunities and limitations in clinical translation. Delivery methods for gene editing. Effective delivery of gene editing components to target cells is a critical challenge in therapeutic applications. Delivery methods can be broadly categorized into viral and non-viral approaches. Viral vectors, such as adeno-associated viruses, AAVs, and lentiviruses, are highly efficient at transducing cells and delivering genetic material. They are engineered to be replication deficient and carry the Cas9 and guide RNA genes. However, viral vectors can elicit immune responses and have limited cargo capacity. Non-viral methods include lipid nanoparticles, LNPs, electroporation, and microinjection. LNPs encapsulate mRNA or plasmids encoding the gene editing components, offering lower immunogenicity and easier manufacturing. Electroporation uses electrical pulses to create temporary pores in cell membranes, allowing entry of the editing machinery. Each method has its own set of advantages and disadvantages regarding safety, efficiency, and specificity. The choice of delivery method is crucial for successful gene therapy and depends on the target tissue and desired outcome. Ongoing research aims to develop safer and more efficient delivery systems. Optimizing these delivery strategies is key to unlocking the full potential of gene editing. Off-target effects and specificity. A major challenge in gene editing, particularly with CRISPR-Cas9, is the potential for off-target effects. These occur when the Cas9 enzyme cuts DNA at unintended genomic locations that are similar, but not identical, to the intended target sequence. Off-target cuts can lead to unwanted mutations, potentially causing cellular dysfunction or even oncogenesis, which is the formation of cancer. The specificity of the guide RNA is paramount in minimizing these effects. Even a few mismatched bases can sometimes lead to off-target activity. Researchers employ various strategies to predict and reduce off-target editing, including careful guide RNA design, using high-fidelity Cas9 variants, and optimizing delivery concentrations. Ensuring high specificity is crucial for the safety and efficacy of therapeutic gene editing applications. Unintended modifications can have serious consequences for patient health. Continuous research is focused on improving the precision of all gene editing tools. Minimizing off-target effects is a key area of ongoing development. Strategies to enhance specificity. To mitigate off-target effects and enhance the specificity of gene editing, scientists have developed several innovative strategies. One approach involves engineering high-fidelity Cas9 variants, such as Cas9 HF1 and eCas9, which have reduced off-target activity while maintaining on-target efficiency. Another strategy is to optimize guide RNA design by selecting sequences with minimal homology to other genomic regions and using truncated guide RNAs. Dual guide RNA approaches, where two guide RNAs are used to target adjacent sites, can also increase specificity. Furthermore, chemical modifications to guide RNAs can improve their stability and reduce off-target binding. Delivering Cas9 as a ribonucleoprotein (RNP) complex rather than encoding it in DNA allows for transient expression and quicker degradation, further limiting off-target activity. These advancements are crucial for moving gene editing therapies safely into clinical practice. Continuous research is dedicated to refining these methods and discovering new ways to improve precision. The goal is to achieve maximal on-target editing with minimal unintended consequences. Ethical considerations in gene editing. The power of gene editing raises profound ethical questions that society must address. One of the most significant concerns revolves around germline editing, which involves making genetic changes to embryos, sperm, or egg cells. These changes would be heritable, meaning they would be passed down to future generations, potentially altering the human gene pool. This raises questions about unintended consequences, informed consent for future generations, and the potential for creating designer babies with selected traits. While somatic gene editing, changes not passed to offspring, for treating severe diseases is generally more accepted, even here, issues of equitable access and potential misuse arise. The debate also includes the ethical implications of editing non-human organisms, such as creating gene drives that could alter entire populations. Open dialogue. Robust regulatory frameworks and public engagement are essential for navigating these complex ethical landscapes. Balancing scientific progress with societal values is a critical challenge. Responsible innovation requires careful consideration of all potential impacts. Somatic versus germline editing. A crucial distinction in gene editing ethics and application lies between somatic and germline editing. Somatic gene editing involves making genetic changes to somatic cells, which are any cells in the body other than sperm and egg cells. These changes are not heritable and will only affect the treated individual. This approach is currently being explored for therapeutic purposes, such as correcting mutations in specific organs or tissues to treat diseases like cancer or cystic fibrosis. Germline gene editing, conversely, involves modifying the DNA of reproductive cells, sperm or eggs, or early embryos. The genetic changes made through germline editing would be passed down to all future generations of that individual. This heritable aspect raises significant ethical and safety concerns, as the long-term consequences on the human gene pool are unknown and potentially irreversible. Most countries currently prohibit or heavily restrict germline editing in humans. The distinction is fundamental to regulatory and ethical discussions. Understanding this difference is key to appreciating the varying implications of gene editing technologies. Regulatory landscape of gene editing. The rapid advancement of gene editing technologies has necessitated the development of robust regulatory frameworks worldwide. These regulations aim to ensure the safe and ethical application of gene editing, particularly in human therapeutics. Different countries and regions have adopted varying approaches, reflecting diverse ethical perspectives and public concerns. For instance, germline editing is largely prohibited in many nations, while somatic gene editing for severe diseases is often permitted under strict oversight. Regulatory bodies like the FDA in the United States and the EMA in Europe play a crucial role in evaluating the safety and efficacy of gene editing therapies before they can be used clinically. These frameworks typically require extensive preclinical data, rigorous clinical trials, and careful monitoring for long-term effects. International collaboration is also vital to harmonize guidelines and address global implications. The regulatory landscape is constantly evolving as the science progresses. Navigating these complex regulations is a significant hurdle for developers of gene editing technologies. Public trust and safety are paramount in shaping these policies. Base editing technology. Base editing is a revolutionary gene editing technology that allows for precise, single base changes in the DNA without creating a double-strand break. Unlike traditional CRISPR-Cas9, which cuts both DNA strands, base editors chemically convert one DNA base into another. This is achieved by fusing a Cas9 nickase, which only cuts one DNA strand, or a catalytically inactive Cas9, decasnoing, to a deaminase enzyme. The deaminase enzyme then chemically modifies a specific base, such as converting cytosine to thymine or adenine to guanine. This targeted chemical conversion results in a permanent base change after DNA replication. Base editing offers enhanced precision and reduces the risk of unintended insertions or deletions, indels, that can occur with double-strand breaks. It is particularly useful for correcting point mutations, which are single base changes responsible for many genetic diseases. This technology represents a significant step forward in the precision of genetic manipulation. Its ability to make subtle, targeted changes minimizes cellular stress and potential adverse effects. Base editing expands the toolkit for correcting a wide range of genetic errors. Prime editing technology. Prime editing is an even more versatile and precise gene editing technology, capable of directly inserting, deleting, or substituting DNA sequences without requiring a double-strand break or a donor DNA template. It combines a Cas9 nickase, which nicks only one DNA strand, with a reverse transcriptase enzyme. A specially designed prime editing guide RNA, PEG-RNA, directs the Cas9 nickase to the target site and also carries the desired genetic edit in its RNA template. After the nick, the reverse transcriptase uses the PEG-RNA's template to synthesize new DNA directly into the genome. This search and replace functionality allows for a wider range of precise edits, including small insertions and deletions, and all 12 possible point mutations. Prime editing significantly reduces the risk of indels and unwanted byproducts associated with double-strand breaks. Its enhanced precision and versatility make it a powerful tool for correcting a broad spectrum of genetic errors. This technology represents a major leap in the capabilities of gene editing. It offers unprecedented control over genetic modifications, pushing the boundaries of what is possible in genetic engineering. Epigenetic editing. Beyond directly altering the DNA sequence, gene editing technologies are also being developed to modify gene expression through epigenetic editing. Epigenetics refers to heritable changes in gene activity that do not involve alterations to the underlying DNA sequence. These changes include DNA methylation and histone modifications, which can turn genes on or off without changing their code. Epigenetic editing tools typically use a catalytically inactive Cas9, decas9, fused to an epigenetic effector protein. The dCas9 guides the effector protein to a specific genomic location, where it then adds or removes epigenetic marks. This allows scientists to precisely activate or repress gene expression without making permanent changes to the DNA sequence itself. Epigenetic editing holds promise for treating diseases where gene expression is dysregulated, such as certain cancers and neurological disorders. It offers a reversible way to modulate gene activity, providing a new dimension to genetic therapies. This approach provides a nuanced control over gene function, offering a powerful alternative to direct DNA modification. It opens new avenues for therapeutic intervention by targeting regulatory mechanisms. Gene drives and their implications. Gene drives are a controversial but powerful application of gene editing that can rapidly spread specific genetic traits through a population. Unlike conventional inheritance, where a gene has a 50% chance of being passed on, a gene drive ensures that a particular gene is inherited by almost all offspring. This is typically achieved by using CRISPR-Cas9 to copy the gene drive construct from one chromosome to its homologous partner, effectively converting heterozygous individuals into homozygotes. Gene drives have significant potential for addressing major global challenges, such as controlling disease-carrying insect populations, e.g. mosquitoes transmitting malaria, or eradicating invasive species. However, their ability to irreversibly alter wild populations raises serious ethical and ecological concerns. There are fears about unintended consequences, potential ecosystem disruption, and the difficulty of reversing such changes once released into the environment. Strict regulatory oversight and extensive public discourse are essential before any widespread deployment. The implications are far-reaching and require careful consideration of both benefits and risks. Responsible development and deployment are paramount for this powerful technology. Future directions in gene editing. The field of gene editing is continuously evolving, with researchers exploring new tools and expanding existing applications. Future directions include the development of novel Cas proteins and other nucleases with improved specificity, efficiency, and expanded targeting capabilities. Efforts are also focused on creating programmable gene editors that can target RNA directly, offering another layer of gene regulation. The integration of artificial intelligence and machine learning is expected to revolutionize guide RNA design and off-target prediction, further enhancing precision. Beyond therapeutic applications, gene editing is poised to play a significant role in synthetic biology, enabling the engineering of organisms for biofuel production, bioremediation, and novel biomaterials. The development of in vivo delivery systems that are safer and more efficient for a wider range of tissues remains a key priority. As technology advances, gene editing will likely become even more precise, versatile, and accessible. The potential for personalized medicine and addressing global challenges is immense. This ongoing innovation promises to unlock even greater possibilities in the coming years. Challenges in clinical translation. Despite the immense promise of gene editing, several significant challenges must be overcome for its successful clinical translation. Ensuring the safety of gene editing therapies is paramount, requiring extensive preclinical testing and rigorous clinical trials to identify and mitigate potential risks like off-target effects, immune responses to delivery vectors, and unintended chromosomal rearrangements. Achieving sufficient editing efficiency in target cells while minimizing effects on non-target cells is another technical hurdle. The cost of developing and delivering these highly personalized therapies is currently very high, raising concerns about equitable access and affordability. Furthermore, manufacturing gene editing components at scale while maintaining quality and consistency presents logistical complexities. Long-term follow-up studies are essential to monitor the durability and safety of genetic modifications over a patient's lifetime. Addressing these challenges requires interdisciplinary collaboration among scientists, clinicians, regulators, and policymakers. Overcoming these hurdles is critical for gene editing to move from promising research to widely available treatments. The path to clinical success is complex, but holds significant potential for patient benefit. Public perception and education. Public perception and understanding are critical factors influencing the acceptance and responsible development of gene editing technologies. Misinformation and ethical concerns can lead to public distrust and hinder scientific progress. Therefore, transparent communication and effective public education are essential to foster informed societal dialogue. Scientists and policymakers have a responsibility to clearly explain the benefits, risks, and limitations of gene editing in accessible language, avoiding sensationalism. Engaging diverse stakeholders, including patients, ethicists, religious leaders, and the general public, in discussions about the future of gene editing is crucial. Educational initiatives can help demystify the science and address common misconceptions. Building public trust is paramount for the successful integration of gene editing into healthcare and agriculture. A well-informed public is better equipped to participate in policy decisions and support responsible innovation. Open and honest dialogue ensures that scientific advancements align with societal values and expectations. Conclusion: Impact of gene editing. Gene editing technologies, particularly CRISPR-Cas9, have ushered in a new era of biological research and therapeutic possibilities. From understanding fundamental biological processes to developing treatments for debilitating genetic diseases and enhancing agricultural productivity, the impact is profound and far-reaching. While significant scientific and ethical challenges remain, ongoing research and responsible dialogue are continually addressing these hurdles. The precision, versatility, and relative simplicity of modern gene editing tools have democratized genetic engineering, making it more accessible to researchers worldwide. As the field continues to evolve, with new tools like base and prime editing emerging, the potential for transforming human health and the environment will only grow. This certificate program has provided a comprehensive overview of these revolutionary technologies. The future of gene editing holds immense promise for addressing some of humanity's most pressing challenges. It is a testament to human ingenuity and the power of scientific discovery.